Karishma for such a nice
Oh my God, please drop the.
Thank you. Mixed feeling. I'm mixed here. A warm good afternoon to everyone present over here, and I'm here to present my thesis work titled Epigenetic Regulation of Skeletal Myogenesis. All our day-to-day activities, whether it comes to walking, climbing, running, or blood circulation, whether it is voluntary or involuntary, depends upon the directional and the coordinated movement provided by muscles. There are three distinct kinds of muscles. They are skeletal muscles, smooth muscles, and cardiac muscles. Among which skeletal muscle is the most dominant and abundant muscle type, accounting for about 50% of the total body weight, and there are about 640 individually typed skeletal muscles. During the process of development, skeletal muscles are established in an elaborately organized manner and sustained throughout life by the process referred to as skeletal myogenesis. Skeletal myogenesis is a multi-step process which begins with the initial commitment of the mesodermal progenitor cell towards the muscle fate. And once when a myogenic precursor cell gets activated, it expands, giving rise to that of a proliferating myoblast, which then fuses together to form a multinucleated myotubes, which then matures to form a muscle fiber. And during this process of skeletal muscle development, there is a coordinated expression of various transcription factors. For example, in the myogenic precursor cells, there is expression of PAX3 and PAX7, which are pair box transcription factors, and they are important for the satellite cell survival and maintenance. Satellite cells are they are actually the myogenic precursor cells. And then once when it gets committed, there is upregulation of MyoD and MIF5, and once when it starts differentiating, MyoD in turn upregulates myogenic, which is an intermediate marker for differentiation. And later, once when it matures to form muscle fiber, there is upregulations of various late muscle specific genes such as troponin T and MLC. And I had used for most of my experiments as a terminal differentiation markers, myosin heavy chain and troponin T, and myogenin as an intermediate markers of differentiation. And in the previous slide you saw there is various transcription factors which has been listed out, among which MyoD, MIF5, myogenin and MRF4 are being broadly classified under the category called myogenic regulatory factors, or simply MRFs. And these are all shares significant sequence homology. And if you overexpress any of these myogenic regulatory factors in a non-muscle cells, let us say in a fibroblast, they are capable of converting such a fibroblast to that of a myotubes. And if you look into the domain structure of either MyoD or MIF5, before going to that, I have to say that MyoD needs to be singled out from this as a master regulator of myogenesis. That's because it is expressed prior to the onset of differentiation, and it is involved in bringing about an irreversible cell cycle exit by induction of P21, and also it upregulates myogenin, which later turns on the muscle-specific genes, thereby bringing about a differentiation program. If we look into the domain structure of either MyoD or any of the MRFs, they have a highly conserved basic domain and helix loop helix domain. And it is through this helix loop helix domain, they heterodimerases with ubiquitously expressed E proteins, and then they bind to the sequence-specific DNA binding elements known as E boxes present on the muscle-specific promoters. And this results in the transcriptional activation of muscle-specific genes. And it's through this basic domains they bind to the E boxes. Although MyoD is expressed prior to the onset of differentiation, and it is capable of bringing about a premature muscle differentiation, this is being checked by means of expression of various negative regulators in proliferating myoblasts. That includes ID, TWIST, MIST1, MyoR, and MyoD. These are some of the transcription factors which keeps MyoD, that alters MyoD's DNA binding ability and its transcriptional activity. However, upon differentiation, these negative regulators are being downregulated, and the positive regulators takes its place, and this results in differentiation. In addition to the negative regulators listed out in the previous slides, there are growing evidences which points out to the involvement of epigenetic mechanism in the regulation of skeletal muscle differentiation. So So what is epigenetics? It is the study of changes in the gene expression or cellular phenotype caused by mechanism other than changes in the underlying DNA sequences. So it is to do with the changes in the histone tail. It's a histone tail modification or a DNA methylation associated with CPGI. So for example, some of the key epigenetic modifications listed out here. Histone acetylation is usually associated with gene activation, and one of the most important key epigenetic modification is to do with H3K9. However, when we talk about histone methylation, depending upon the lysine residue which is subjected to methylation, it can be either a gene activation or gene silencing. For example, here H3K9 methylation results in transcriptional repression or is gene silencing. However, H3RA, which refers to histone 3 or G9 at position 8, when it is methylated, it undergoes gene activation. So this sort of histone tail modification, whether it is methylation or acetylation, is brought about by a broad spectrum of enzymes, collectively called as chromatin modifying enzymes, under which we have histone lysine acetylation, which is catalyzed by histone acetyl transferases. It comes under Hatch family that includes CVP, P300 and PCAP. And histone lysine B acetylation is catalyzed by histone B acetylases, which are broadly classified under class 1, class 2 and class 3. And finally we have histone lysine methylation catalyzed by histone lysine nucleic transferases that includes U39H1, H2, G9A, ZDB1 and ZDB2. And later I will be talking about G9A. Now coming to a skeletal muscle system, in an undifferentiated myoblast, if it takes a muscle promoter, it undergoes a closed chromatin configuration. This is due to the association with that of many chromatin modifying enzymes such as histone nucleic transferases, which brings about a key epigenetic modification here at H3K9, which undergoes either mono, di or trimethylation. And this is because of the association of MyoD with Su39H1, which is the histone nucleic transferases, and its association with histone D acetylases. This results in MyoD becoming transcriptionally inactive here, and thus it blocks the myogenic differentiation program. However, upon differentiation, these repressive chromatin marks are replaced by active chromatin marks such as H3K9 acetylation, that is because of the association of MyoD with acetyl transferases such as PCAP and P300, which directly acetylates MyoD and thereby drives the myogenic differentiation program as a consequence of open chromatin configuration. Our lab has previously shown that SHARP1, which is a VHLH transcription factor, is expressed in skeletal muscles, and overexpression of SHARP1 results in blocking the myogenic differentiation program. There was a second paper to follow this from our lab, which showed that SHARP1 inhibits adipogenic differentiation, and here, interestingly, we found that it retains the co-repressor G9A on the adipogenic promoters, and thereby blocking the adipogenic differentiation program. So we thought, how about SHARP recruiting G9A on the myogenic promoter, thereby blocking skeletal muscle differentiation, and thus forms the hypothesis to investigate the role of G9A in skeletal glycolysis. So what is G9A? G9A is a eukaryotic histone lysine N-methyl transferase 2. It's a major H3K9 nucleotransferase which is responsible for the transcriptional repression of many euchromatin genes. And previous studies have shown that loss of G9A in mice results in growth retardation and early embryonic lethality. If you look into the domain structure of G9A, it has an ankyrin repeat domain and a Z domain. Ankyrin repeat domain is implicated in protein-protein interactions, whereas if you look into the Z domain, it is through the Z domain, it transfers the methyl groups to the H3K9, resulting in the transcriptional repression of many euchromatin genes. In addition to its role in mediating the H3K9 dimethylation, it has also been shown to mediate methylation of non-histone substrates, and some of the major transcription factors that are methylated by G9A includes P53 and CBP beta, which are being negatively regulated as a result of such methylation. So thus comes my first aim, that is to investigate the role of G9A in regulation of skeletal muscle differentiation. To analyze this, to carry out the experiment further, I was using C2 sequel cells, which is a mouse myoblast cell line, and it is a useful and easy model to study the process of skeletal muscle development. So when we take C2 sequels in a growth medium, that is in an undifferentiated state, it looks like this. And under low serum condition and under higher confluency,